Hi, my name is Michael Silverstein, and I'm excited to tell you about my PhD research, which involved studying the principles of microbiome structure and the implications of those principles for climate change mitigation. Some of you may know, greenhouse gas emissions are increasing the rate of climate change, greenhouse gases such as carbon dioxide. Something I didn't learn about until my PhD was about how some of the carbon in dead microbial cells can associate with soil minerals. And when that happens, that carbon can be stabilized in the ground for hundreds to thousands of years, providing a natural mechanism by which we can store carbon. The more I thought about that, I started to consider, could we perform microbiome engineering to increase the rate of this carbon stabilization? And the more I thought about that, I started to think about, what does it take to perform environmental microbiome engineering at all? In order for us to understand what microbiome engineering is, the first thing we have to do is understand what a microbiome is. Well, a microbiome is an interacting community of microorganisms in an environment. And they're comprised of taxa, that's who is there, such as bacteria, archaea, and fungi, organisms like that. And these organisms do all sorts of things, such as consuming resources, like this bacteria here that just consumed this resource. And once inside of the cell, enzymes within these organisms can transform resources into new ones. Now, these organisms may use these new resources for themselves, or they may even secrete some of these back into the environment where they're cross-fed to other organisms. Microbes also compete for resources. Now, these are just some of the things that microbes do, but you might be wondering, how is this all related to climate change? One of the main ways that microbiomes are related to climate change is through metabolism and the carbon cycle. For example, we have this resource over here on the right where I've labeled a carbon atom in this resource in red. Now, some microbe might eat that resource, and that carbon atom may end up in the biomass of that microbe. However, that same carbon atom may end up in carbon dioxide and be respired back into the atmosphere as a greenhouse gas. So here you see how the metabolism of the microbes in a microbiome are directly linked to climate change. To understand microbiome engineering further, I'll discuss the three sections I've outlined here. In the first one, I'll go into more detail about the definition of microbiome engineering. In the second section, I'll discuss the divergence-complexity effect, which is an ecological phenomenon I discovered, which has to do with how microbial communities assemble in different ways in environments of varying metabolic complexity. And then in the final section, I'll discuss functional response, which is a way that we can relate the differences in taxonomy, who is in a microbial community, with function, what is happening in those microbial communities. So to start off, we'll further explore the definition of microbiome engineering. What I'm showing here is what I refer to as the microbiome engineering process, and this is the result of an extensive literature review that I performed. Starting all the way on the left is the first step of the process, where you have to identify an ecosystem function target of interest. This can be a metabolite you want to be consumed in the environment, some compound you want to be released into the environment, the production of a certain enzyme, increasing the growth of plants, whatever it might be. This is the thing that you want to change in the environment that microbes regulate. The second step is to source some microbes. This can be gathering isolates or entire microbial communities. Once you have your microbe, the next step is to design what I refer to as an inoculum community. That's what's being shown in the middle in orange. The reason why this is called an inoculum community is that this community will be delivered or inoculated into some environment, which in this case is represented by the blue rectangle in step 4. Now, this is an environmental community already resides in this environment, which I'm showing in the blue circle. So in step 5, the final step of the microbiome engineering process is the monitoring of establishment. Here, what we're hoping to see is that our inoculum community in orange coalesces or mixes with the resident community to lead to a resulting community that has increased activity of our function of interest. Now, there's many questions we can ask about the different steps in the microbiome engineering process, but one of the first questions we might want to ask is, how can we increase the rate that carbon is stabilized, since this could help us with climate change mitigation? Now, I'm going to propose that a good target for that is something called carbon use efficiency. To define carbon use efficiency, we'll start off with carbon dioxide, which I'm showing over there on the left. Now, you may know that plants can consume carbon dioxide, and when they do that, they may use that carbon dioxide to create a sugar, where that carbon atom that used to be in the atmosphere is now in that sugar. Now, this plant may secrete this sugar into the soil, where it could be consumed by some microbe. And now that carbon atom that used to be in the atmosphere and was in that sugar is now inside of a microbe in the soil. Now, if that carbon atom ends up in a particularly dense part of the cell, such as the cell wall, when that microbe dies, that cell wall component may form a really strong association with soil minerals where it'll be stabilized for hundreds of thousands of years. Or that carbon atom could be re-respired back into the atmosphere. So from the perspective that an efficient use of carbon is to generate biomass, the carbon use efficiency is the amount of carbon that ends up in biomass divided by all of the carbon in the system. Now, some really excellent work was done last year, this massive meta-analysis of 57,000 different soil samples. And what they're showing on the x-axis in these plots on the left is the microbial carbon use efficiency, and on the y-axis is the amount of carbon in the soil. And as you see, soils with a higher carbon use efficiency have more carbon in them. So what this suggests is that if we could increase the carbon use efficiency of microbial communities, this could lead to more stabilized carbon, which could lead to a negative atmospheric flux of carbon from the atmosphere into the ground, which would help climate change mitigation by removing these greenhouse gases from the atmosphere. So in order to easily study carbon use efficiency, we want a way of measuring carbon use efficiency in high throughput. So to quantify the carbon use efficiency of a community, we need to be able to measure biomass and respiration. So one thing we could do is we could take soil, and then we could put soil into a 96 well plate, which I'm showing here, where each well has a different soil sample. What's really nice about this 96 well plate is that it can be used for high throughput measurements. For example, we could put this plate into a plate reader or a spectrophotometer, which I'm showing here, where you can see this little tray that's sticking out of this device can hold the 96 well plate. And what this device does is it measures the optical density of each of those wells, and we can then use that to approximate the biomass of each of these samples. Also in a 96 well plate format, we can measure the respiration. And one way we could do that is with this device here that I'm showing, which is called a microresp. So here you seal a 96 well plate with another 96 well plate that contains this purple gel, and this purple gel reacts with carbon dioxide. And when it reacts with carbon dioxide, it changes the color of this gel. That plate where the color has changed the gel can then be placed back into a spectrophotometer, where you could then measure the amount of respiration that has occurred. So I have done some preliminary validation of this method, which I'm showing the results here, where on the x-axis of each of these plots is the results of one replicate, and on the y-axis are the results of another replicate. And each of these subplots is measuring either the respiration, the biomass, or the carbon use efficiency of each of these replicates. And we were happy to see with this very preliminary validation that we have decent correlations between replicates. And what this suggests is that using optical density and the microresp could be a promising approach to measuring carbon use efficiency in high throughput. So that was all about ecosystem function targets. And another question we could ask is about how can we actually go about designing an inoculum with maximal carbon use efficiency? And I'm going to propose that one of the best ways we can do this is with a technique called directed evolution. There are many different ways to design inoculums. For example, we could design inoculums from the bottom up, like using a single organism, which is really common in agriculture to boost plant growth. Or in panel B, I'm showing an assembled consortia, where different isolates can be put together to form a small community. We could also perform synthetic biology to introduce certain genes into either a microbe or into a community. We could also employ top-down approaches. One example is to use a whole community, like in panel D. This is the case with fecal transplant, where an entire microbial community from a healthy person is introduced to a patient. We could also design an enriched consortia, where a whole community is enriched or cultured on some growth condition where certain microbes in the community are selected. Or we could perform directed evolution, which I'll be explaining in the following slide. So before telling you about how directed evolution works, the first thing I need to describe is the community function landscape, which I'm depicting here. So imagine that this is a hypothetical community of a yellow and blue taxa, where the x-axis represents the abundance of the yellow taxa and the y-axis represents the abundance of the blue taxa, and each point in the plot represents the carbon use efficiency associated with each community of yellow and blue taxa in these different abundances. So you'll notice that for most communities of the yellow and blue taxa, the carbon use efficiency is this blue color, which if you look at the color bar on the right, represents this low carbon use efficiency. However, there are some points in this landscape where the carbon use efficiency is much higher for certain communities. So the goal of directed evolution is for us to be able to explore this landscape and try to find these points of higher carbon use efficiency. Now, another important component of the community function landscape is the idea of stable points, which I'm representing with these x's here, these red x's here. Now, these stable points are combinations of the yellow and blue taxa that are stable. So if you have some community that deviates from one of these stable points, it'll be attracted towards one of these stable points. So here is how directed evolution actually works. So let's say we want to try and find the maximum point in this landscape. The first thing we could do is start off with some random combination of yellow and blue, such as the community that's described by the white point in the middle of the plot right now. So the first step of directed evolution is, once we have started with a community, is to perform a community perturbation. Now the purpose of this is for us to explore this landscape, for us to try a new combination of yellow and blue. Now there are many different ways to perturb a microbial community. You could dilute the sample, you could inject more yellow or blue taxa into the sample, you could introduce new nutrients, you could change some environmental parameters such as the temperature. Now there's many things you can do, but the point is that we're somehow changing what this community looks like. Following this perturbation, we will then stabilize towards the nearest stable point. At this point, we can ask the question, have we found a community with a higher carbon use efficiency? In this case, the answer is no. We're still in this sea of blue. So since this is the case, we can perturb again and then allow the community to stabilize again. And now we can ask the question again, did we find a point of higher carbon use efficiency? Which in this case, the answer is yes. So in this way, directed evolution is sort of like this biological optimization problem that's very much inspired by natural evolution, where we have these cyclic iterations of mutation and selection till we find sort of more fit configurations. So we don't need to perform directed evolution with just one community. We could initialize with multiple communities and then simultaneously perform directed evolution. And in this way, we can comprehensively explore this landscape to try to find points with higher levels of carbon use efficiency or any type of function that we're interested in. So that concludes my discussion on the first chapter of my PhD research, where I talked about microbiome engineering. And just as a summary, some of the things I spoke about was how carbon use efficiency can be a great target for carbon stabilization and downstream climate change mitigation. I talked about how optical density in the microresp could be used as a high throughput measurement for carbon use efficiency. And I also told you about directed evolution as a technique for designing inoculum communities. So up until this point, I've focused on identifying ecosystem function targets and strategies for designing microbial inocula. And now for the remainder of the talk, I'll be investigating this question about establishment, the final part of the microbiome engineering process. Specifically, I'll be asking this question here about, is it the case that some environments may be more susceptible to microbiome engineering than others? And to address this question, I'll be introducing you to this concept of divergence. And to start off, I'll be talking about this ecological phenomenon I discovered called the divergence-complexity effect. To understand the divergence complexity effect and to understand how it relates to microbiome engineering, I'll first start by exploring this hypothetical thought example here about how many states might be available in different types of ecosystems. So once again, I'm showing this community function landscape, and in this case, there's only one stable state here, as denoted by this red X. Now, if this is the case in an ecosystem, then there's no way for microbiome engineering to be able to work in this ecosystem, because any attempt to deviate from this stable state will fail because the community will just be attracted back to the stable state. So there will be no way to change who is there or what is happening in this ecosystem. Now, this is particularly clear when comparing the ecosystem on the left to the ecosystem on the right, where now there are many stable states. And maybe it's the case that we're starting at some community like the one that's marked with the white dot, but maybe we can move to a new state that is just as stable as the initial one, but exhibits a higher level of function. So this thought experiment got me to ask the following question about what is it that determines the number of microbial communities or states that an environment can host? Now, to investigate this question, the first thing I did was to perform a meta-analysis on two different studies that assembled microbial communities in different conditions. The first study assembled soil microbial communities in a very simple environment that just had glucose as a carbon source. Another study collected microbial communities from pitcher plants and cultured those communities in an acidified cricket media, which is meant to represent the environment that these communities came from, and is notably far more complex than just glucose. Now, to properly compare these experiments, I wanted to reprocess the raw data so that differences I see between the studies are due to biological differences and not just technical ones. So let me really briefly describe to you how we acquire community data, and then I can tell you what I did to reprocess this data in the same way. So microbial community data starts with a microbial community where DNA is extracted from that community, and that DNA is then sequenced. And the result of that sequencing is what we call the raw reads, where each of these reads is a string of nucleotides, those A, T's, G's, and C's, and each of those reads is coming from some taxa in your community. These raw reads can then be processed where each of these reads are essentially counted, and what you end up with is the relative abundance of each taxa from your original community. So, this data is such a common task, I figured that it would make sense to make it available on BU's shared computing cluster. So what I've done is I've developed this tool called BU16S, and this pipeline lives on the shared computing cluster, and all that's required for using it is to enter module load BU16S. It has batch processing capabilities, it allows for multiprocessing, it produces logs too so that researchers can keep track of the different types of processing that they've done. BU16S has already been used in 3 different publications and by multiple people in the BU community. So I use BU16S to process the data for my meta-analysis. What I'm showing here are the first results from this meta-analysis of the soil communities being grown on glucose. Each of those lines of a different color represents a different microbial community, and the farther away different points are means that those communities are more different in taxa. So they're more different in terms of who is there between those different communities. The circles represent the initial soil communities. So what you can see is that initially these communities are very different from each other, and from each circle is a line with an arrow, and that shows how the community changes over time. The squares represent the final community composition. So what it looks like in this very simple environment, the communities start off very different from each other and they become closer and closer to each other over time. This is a very different story than what we saw in the pitcher plant communities. So these pitcher plant communities do start off very different from each other, but these final points diverge much more strongly from each other than the ones in the glucose conditions. In fact, it almost seems as if some of these pitcher plant communities are arriving at different stable states, where you see that some of these squares are clustering together and other squares are clustering together in other places. So when I saw it led me to believe that maybe between these two different conditions lies a latent gradient of metabolic complexity, and that in simple conditions, there's very little divergence, and in more complex conditions, there's more divergence, leading to this general hypothesis that maybe complex environments lead to more divergence. But in order to test this, I needed my own controlled experiment. So that's what I did. I went out into the woods, I collected 6 different soil communities and brought those back to the lab and grew those communities in 9 different conditions of increasing metabolic complexity. The different metabolites I used for my conditions are being shown here: citrate, glucose, cellobiose, cellulose, and lignin, where these metabolites increase in complexity from left to right. 5 of the conditions I used were single metabolite conditions using the metabolites I just described, and in this way we can define metabolic complexity by the chemical complexity of each of these metabolites. I also used some mixed metabolite conditions. Citrate glucose was one, citrate glucose cellobiose was another, etc. There are 4 different combinations like this. And in this way, we were able to define metabolic complexity by the diversity of metabolites in the condition. So I grew those 6 soil communities in these 9 different conditions for 30 days, passaging every 3 days. And then I performed 16S sequencing, which tells us who is in each of these samples. And then I used BU16S to process this data. Now, the first result that I'd like to show you from this experiment is from the citrate condition. This is our simplest condition. And this is the same type of plot that I showed before, where initially we see that the communities are very different from each other, represented by those circles. And over time, we see that the soil communities growing in citrate become very similar to each other. Those squares are very close to each other. This is especially clear when we compare our simple condition to our most complex single metabolite condition, lignin. Initially, the communities are just as different from each other, but now we see these communities diverge much more strongly from each other in their final state. In fact, it almost looks like these communities are arriving at different stable states. Now, this is a great qualitative way to look at the data, but we can also quantify this divergence. So in this y-axis here, the higher we are in the y-axis, the more divergence, the more distance there are between those final states, those squares in the little plots. And the lower we are on the y-axis, the closer the final states are to each other. And here we see in this quantitative representation of how the lignin condition, the communities in the lignin condition diverge more from the communities in the citrate condition. Now I'm going to show you the results from the remainder of our conditions. So in the blue boxes are our single metabolite conditions. In the orange boxes are the results from the mixed metabolite conditions. And in each of these different situations, as the colors go from light to dark, the complexity of the conditions are increasing. And by both definitions of complexity, we see that communities diverge more in the more complex conditions, which led us to identify the divergence complexity effect, where we observe that the divergence of communities increases more with the metabolic complexity of their conditions. Now, this was extremely exciting for us. This perfectly aligns with our hypothesis. However, we didn't want to stop at just making this observation. We wanted to understand, why does the divergence complexity effect exist? Now, there were two hypotheses that we came up with for why we might observe the divergence complexity effect. The first one is that it is a result of some physiological effect. And I'll show you what I mean with this example here. Imagine we have the two hypothetical communities I'm showing on the left, and imagine that both of them have some fast-growing taxa, this red one. Now let's imagine we grow these communities in a very simple condition, and we could imagine that this fast-growing taxa would dominate these two different communities. So although these two communities looked different initially, now that they've been dominated by this fast-growing taxa, they look much more similar. So the first hypothesis is that fast growth in simple conditions drives convergence. Another hypothesis is that the divergence complexity effect is driven by an ecological effect. So using those same two communities, we can imagine that this purple enzyme is shared between the two communities and can cleave this linkage here where the enzyme is being shown. But only community 1 has this yellow enzyme, which would mean that community 1 would have this specific byproduct, this specific polysaccharide that community 2 doesn't have. And by having this different polysaccharide, community 1 could experience a whole downstream cascade of resource transformations and resource availability that community 2 does not experience. And this difference could be a way in which community 1 and community 2 ultimately assemble differently. So this second hypothesis is that different specialists in complex conditions drive divergence. This is really about differences in physiology between individuals, whereas the second hypothesis is really about differences between interactions, this ecological effect. So in order for us to test the ways in which these hypotheses might be involved with the divergence complexity effect, I use something called a consumer resource model to perform some simulations. So a consumer resource model consists of two components. There are resources and consumers. Resources are shown here as these circles, whereas the consumer is this bacteria-looking icon. In this model, you can define consumers by which resources they prefer. So in this case, we could define this blue bacteria as a consumer that prefers blue resources. Resources are defined by how they transform. In the example on the left, this dark blue resource is becoming this dark orange resource. And with these two things, we can then have all sorts of ecological phenomena, such as cross-feeding and competition, emerge. Now, since this is a computational model, we get to choose how the resources and consumer preferences are structured. A natural choice is to use what I will refer to as a trophic structure. Trophic meaning different levels. So in this case, there is a blue level and an orange level to this ecosystem. And there's a unidirectional flow of energy that goes in a hierarchy from complex resources and the consumers that prefer those complex resources to simpler resources, the orange resources. So I'll be referring to structure and consumer preferences as trophic consumer preferences, and I'll be referring to trophic structure and resource transformations as trophic resource transformations. So with trophic consumer preferences, you'll have groups of specialists that prefer a group of resource types, and in trophic resource transformations, resources will transform from complex resources towards simpler ones. Now, in order for us to test the effect of this trophic ecological structure with the divergence complexity effect, we need a null model to compare to. A natural choice here is to have a model with random structure, with random consumer preferences, where there's no sense of groups of taxa that prefer groups of resources anymore. Taxa can just prefer any old resource. And we can also have random structure in resource transformations, where there's no sense of complex resources transforming into simple ones anymore. So using these combinations of trophic and random parameters, there are 4 different models that we can construct. The first one we can construct is one in which we have trophic consumer preferences and trophic resource transformations. We can also have trophic resource transformations, but with random consumer preferences, trophic consumer preferences, but random resource transformations, or random of either consumer preferences or resource transformations. Now, in each of these cases, I maintain the same growth, utilization, and death rates across all consumers. And in this case, we're controlling for the physiological parameters between all consumers, meaning any differences that we see are due to differences in ecological interactions. So with these simulations, we're really testing the ecological structure. So if we're able to reproduce the results that we saw from our experiment, this suggests that ecological structure is strongly implicated in the divergence complexity effect, which was one of my hypotheses. So for each of these different models, I performed simulations, and then with each of the resulting communities, I computed the divergence, and then I compared the divergence of these simulated communities with what I measured in my experiment. And the tools for performing these simulations are available on GitHub at the link shown here. So just as a reminder, on the left is our experimental result of the divergence complexity effect, where increasing metabolic complexity leads to more divergence between communities. And what I'm showing on the right there in panel D is the first result from our consumer resource model simulations, where we see with random consumer preferences and random resource transformations, we do not reproduce the divergence complexity effect. Now, this is somewhat expected because this is the case where there's no sense of structure at all. So it's a bit reassuring to see that we don't see our effect in this scenario. Now what I'm showing you is the remainder of the results in using our other models. And what immediately might jump out to you is how in panels A and B, in that first row there, we see results that very strongly reflect what we saw in our experiment. Now, if we were going to reproduce our results in any case, panel A is the most likely candidate because this is the case where we have both trophic consumer preferences and trophic resource transformations. Reproducing the results in panel B was a little bit more surprising because here we have random consumer preferences. Not producing the results in panel C was also surprising because there we have random resource transformations. So what this suggests to us is that the hierarchical resource transformations are what allow for the divergence complexity effect and these differences in assembly, because it's in the cases where we have trophic resource transformations, even when consumer preferences are random, we're able to reproduce the divergence complexity effect. Now, I won't be going over the remainder of the analyses we performed in our study today, but I just wanted to show you how in this top row here, we have other results from our experiment. And in the bottom row, we have the corresponding results from our simulated communities. And we're really happy to see a strong correspondence between our experimental and simulated results. So just in summary about this chapter here, I've introduced you to the divergence complexity effect, which is summarized in the schematic in the middle of the screen right now, where on simple conditions, different communities, like in panel B, will converge. However, those same communities in a complex condition will diverge. I've shown you how, with our consumer resource models, that this effect is driven by hierarchical resource transformations. And what's exciting is that all of this leads to a new hypothesis that maybe it's the case that complex environments which lead to more divergence may have more states available for communities to assemble to in an ecosystem, which maybe suggests that it's complex environments that are most susceptible to microbiome engineering. So that concludes my discussion about chapter 2, And next, I want to talk to you about functional response, which critically relates differences in taxonomy to differences in function. After I talked about taxonomic divergence, which I'm depicting here with these three communities that arrive at different stable states. Now, this is great, but if we want to successfully perform microbiome engineering, what we want to do is change who is there, so move between these different stable states in order to change what is happening. For example, maybe we would start off at one of these stable states on the left, and in the middle, what I'm showing is where this community exists in functional space. And our goal is to be able to change what's happening in taxonomic space, change who is there, and for that to lead to changes in what's happening in that community. So if we successfully reach this target and change what's happening, that's a success. However, you could imagine that we could change who is there, but then not have a corresponding change in what is happening in these communities. So the prospect of these two different scenarios occurring led me to ask this question of when do differences in taxa translate to differences in function? Now, to explore this, we can look to a really common example where different microbial communities were sampled from a bunch of different plants, these bromeliad plants. And each of the columns in this plot on the left here represents the different taxonomic profiles of each of these different plants. Now, when you look at all these bars, they look totally crazy, totally different from each other. There's really high divergence taxonomically between these different communities. Now, with those same exact plant communities, when the researchers looked at the functional profile, so what's happening in those communities, the columns look far more similar to each other. They diverge far less from each other. Now, this would be really challenging for microbiome engineering because in this ecosystem, you can change who is there, but what this suggests is that there's very little differences in what is happening between these communities. This is a little bit similar to how you could imagine that there's different types of people between a city like New York and Boston. New York and Boston could look really different in terms of who is there, but these cities might have really similar things. They have musicians and cobblers and butchers and lawyers and people that sell pizza. So seeing this in one ecosystem raises the question, are all ecosystems like this? And this really necessitates the need to have a way to relate these differences in taxonomy, the differences between communities in terms of who is there, and differences in what is happening functionally. So to understand the relationship between taxonomy and function in ecosystem, I developed a metric which I'll explain to you now. And to do so, I'm going to go through a hypothetical example. So imagine that we have two communities, C1 and C2, which are comprised of different amounts of a yellow, blue, and purple taxa. What I'm showing in panel B is how we can then represent the differences between C1 and C2 in this taxonomic space. And then, of course, we can then measure the distance between C1 and C2, like I've shown there with the arrow. We can then consider how C1 and C2 look in a functional space by considering the genomic content of the yellow, blue, and purple taxa. For example, we could look at which enzymes the yellow, blue, and purple taxa have. We could then use these genomic profiles to map the abundance of C1 and C2 from a taxonomic space in panel B to a functional space, which I'm showing in panel D, where now the x-axis represents the abundance of the green enzyme and the y-axis represents the abundance of the purple enzyme. Now we could do this same exercise for other communities like C3 and C4, which I'm showing in gray. We could then take the pairwise distances between each of these communities and plot them in this plot that I'm showing in panel E. So in the x-axis in panel E is the taxonomic distance between each pair of communities, and on the y-axis is the functional distance between each pair of communities. We can then draw a line between each of these pairwise distances, and this will give us what I call the functional response. Now, the functional response is a single metric that quantifies how much a community functionally responds to changes in taxa. As you move on the x-axis in panel E, you're looking at increasing taxonomic distances, and on the y-axis is literally showing you how much difference in function corresponds with those changes in taxa. So in this example here, that solid black line represents a positive functional response. And this would be a great scenario for microbiome engineering because here, large changes in who is there leads to large changes in what's happening. The dashed line refers to this redundancy case where you can have really different communities in terms of who is there, but no differences in terms of what's happening functionally. Now, this has all been an example on a hypothetical scenario, but of course we can look at real data, like the example I'm showing here, which are from a bunch of different communities in a marine system. Now, this is just one study. However, we can look at a whole bunch of studies. What I'm showing here are 33 different environmental studies from the Earth Microbiome Project. Each of these subplots shows studies from a different ecosystem, and each line represents the functional response from a different study within that ecosystem. Red lines represent statistically significant functional responses, whereas the blue lines represent the redundancy case where there's no difference in function with change in taxa. Now, this is a little bit challenging to look at, so what I've done here is I've taken the slope or the functional response of each of those lines, and now I'm representing each of those as a bar, where each of the colors represents a different ecosystem. Now, what's nice about this quick meta-analysis that I've done here is that we have, in some cases, multiple studies per ecosystem. And this is a nice way to validate our metric by looking for, are there instances in which there is consistency among different studies within the same ecosystem. And we see an example of that here with the orange bars, where these are different soil communities that tend to have a positive functional response, and with the purple bars, which are animal gut studies from which are different studies from the animal gut, where we mostly see a negative functional response in those ecosystems. Now, this is nice to see, but one of the challenges with using environmental samples is that there's no way to know if the differences that we see between samples are due to differences in the environments those samples were taken from, or differences in the communities. And what I mean by this is you can imagine going into a forest and taking from very near each other two different soil samples that are really different environmentally. And those environmental differences could be chiefly responsible for the differences that we see here. So what this suggests is that we need to have a controlled experiment to validate this functional response metric. An experiment where different samples are grown in the exact same conditions. Only this way will we be able to test if communities can display a functional response within an ecosystem. So in order to do this, I worked with Sarah Bald, who is a lab mate in Daniel Segre's lab, and we went out into the forest and we collected 10 different soil communities. And then we grew these soil communities in 10 different conditions, some organic acids, some amino acids, some sugars, and in some complex media. We opted to use these different conditions because of an excellent study that came out this year that shows that bacteria tend to prefer either sugars or acids. So by using conditions that have acids and sugars, we were hoping to capture a wide range of responses from these different communities. We grew those communities for a few days, and then we performed 16S sequencing to quantify who was there, the taxonomy, and also performed shotgun metagenomic sequencing, which enables us to look at what different functions are occurring in these communities. And we already have some great preliminary results, which I'm showing here, where on the x-axis are our different conditions. The yellow boxes here represent taxonomic distances, and the blue boxes represent functional distances. And we are happy to see that these distances cover a wide range on the y-axis, and that there appear to be differences between our different conditions. However, there's more analysis to be done with these data. So I'd like to wrap up by speaking to the ultimate goal of this research, which could be thought of as an autologous soil microbiome inoculum for climate change mitigation, where autologous means that we're going to be performing a transplant from an ecosystem back into that same ecosystem. So you can imagine that for a given forest, we could source multiple soil samples and then find the samples with the highest carbon use efficiency, and then even improve that further by performing directed evolution. And then, because my research suggests that complex environments like a forest are more susceptible to hosting communities of different states, we could then reintroduce or inoculate this directly evolved community back into this forest with the hopes of raising the carbon use efficiency of this forest using completely native species, just arranged in a new way that have a higher carbon use efficiency. In conclusion, there's a few things that I've spoken to you about today. The first was about using directed evolution to increase carbon use efficiency of microbial communities and how those communities could be used as an inoculum to boost the carbon stabilization of forest soils. I also told you about the metabolic complexity effect, which suggests that microbial communities are able to diverge more in increasingly complex environments. I also told you about functional response, which is a new metric to be able to understand how changes in taxonomy relate to changes in function and how we see that they actually vary between different ecosystems. And in conclusion, my hope is that microbiome engineering has the potential to mitigate climate change and provide control over other ecosystem function targets. Thank you so much for your time.